Yeah, hope that I am in life. So today I'll just go through about the some CSIR important topics and hope that you guys can go through this topic and hope that that will uh, help your examination. So like and some of the important aspects of that specific topic and you can go through that. So and some questions will be from that specific region. So let's talk about, and last day in that live session I talk about some of the human genetics, whether they are pedigree and how that can how this kind of questions are coming. Already I have discussed on that the human genetics part, but still I think another important aspects is the gene interaction. So that is one of the key question region. Like you can check out the in the gene interaction portion. Lot of questions are coming because few things are more important, and also some other type of genetic informations are there. So today I can discuss about some what I can discuss. Okay, let's discuss about the some molecular marker. So because in your research time or in your research lab, most of the cancer research lab are working on the molecular marker, and along with that some those are working with some plant science or agricultural science. That time also the molecular markers are important, not the whether I am working on the cancers. That's why I more talk about the cancer biomarker or cancer marker, but molecular marker are more essential when you talk about the biotechnology, genetic engineering. In that aspects, it's more essential. So in that live video, live session, exactly I don't know. Technically, here no people in that live because the reach is not too much. But still, I am just going to that more details. In after that, if you check that and hope that this can be your useful, because this time you guys are taking nap after that. So yeah, I'm just want to remind those things, and that can be useful to remind all of you who have studied in previous 1 year or 2 year or 3 year. So firstly I'll go through the few basics of like RFLP and AFLP and the STR along with the SNPs and the STS. So these are kind of mutations. You can They are more common type of mutations, and most of the research are going on in a molecular biology sense. They are SNP research and along with the RFLP research and AFLP research. So let's talk about the SNPs first, like single nucleotide polymorphism. It's kind of that the site whether they are single nucleotide is polymorphic among the all different individuals, like single molecule, single nucleotide base pair is getting changed, and for that changing their expression of the protein get changed. That's the thing. So that kind of research is kind of SNP research. And when you talk about of the RFLP, now it's going to the RFLP, like restriction fragment length polymorphism is kind of the difference in their homologous DNA sequence that can be detected by the presence of fragments of different length after digestion of the DNA sample in the specific restriction endonuclease. That means you take some of the homologous DNA sequence. You know about the homologous DNA sequence. That means their every sequence is kind of same in same loggy. So that's why. But when you digested that, in that the length getting different. So that is the RFLP. And what is the AFLP? AFLP is like the amplified restriction fragment length polymorphism. So first term very important, like amplified restriction fragment. Amplified restriction fragment length polymorphism, kind of DNA fragments of 80 to 500 base pair that often form a digestion with the restriction enzyme followed by the ligations of the oligonucleotide adapter to the digestion provided by the pre-amplification or selective amplification by the PCR. So that kind of AFLP. And another two types I have already shared the term, the STS, like sequence tagged sites, is a general term, describes that any kind of molecular marker that can found the unique site of the genome is SNP and can provide that the sequence tag site within a genome. That kind of the STS site. And you can say about the STR site, like STR, it means they are short tandem repeats, that kind of the micro satellite of that pattern of kind of 6 nucleotides that can be repeated directly, that adjacent to each other. It's kind of the length, usually it will be like 100 to 150 base pair of that length. And this kind of polymorphism occurs when the homologous STR, like tandem repeats, is lossy differ from that number of the repeats between the individual. So these are the things, or these are the molecular marker essential for the for you can say for exam purpose, and along with the, if you want to engineer the cell, at that time you should think and take care of about that. And also for the disease cases, these are also important. For more specifically in the SNP region, lot of mutations are kind of point mutations, and for that point mutation the protein, whatever protein, it's the kind of alter structure protein is formed. And if alter structure is protein form, and that can generate the cannot generate the proper function of that specific protein, and that is the one of the major concern. Now how you can check out that if you take out some of the DNA samples, firstly when you talk about the RFLP, and how do you analyze the RFLP of a chromosomal DNA of 3 kind of individuals. Let's take the individual 1, individual 2 and individual 3. So what you can do it? So you can do like they are take the same individuals and digest it with the same endonuclease. Like say EcoRI, you can digest with the EcoRI, cut side of EcoRI. So like individual 1, individual 2, individual 3. You take the DNA sequences and DNA like, and also cut with the same restriction endonuclease. So what you can get? You can get that the in lane 1 you get the 5000 base pair, one band is there, another is 3000 band is there, another is 2500, 2000, and one is the 1500 base pair. So you just your base pair in the ladder, you are not using ladder, you just digest it with the EcoRI endonuclease, and you get the 5000 band, and you get the don't get the 4500 band, you get the 3000 band, 2500 band, and 2000 band, and the 1500 band. So let's the individual 1, and in the individual 2 you found that like 5000 base pair band, you get the 4500 base pair band, but still get another 2-3 band in between the 4500 to 3000, but you don't get any band from the 3000. And along with the same band, like in 2500 you get the same band, you get the same band of 2000. Now you talk about the individual 3. In the individual 3 you get the 5000 band, 4500 band, 3000 band, 2500 band, 2000 band, along with the 1500 band. So all the band you get that after the digestion. So in first cases you don't get the 4500 band, but in the second cases you don't get the like 3000 band, but in the third cases you get the all the band. So now you can calculate those things. They like that you have missed in the second individual 1500 band is not there, and along with that the 3000 band is not there. But in 4500 you get a in third individual you get the all the bands. In first individual you get missing the 4500 band, but in second individual you missing the 3000 and 1500 band. That means these 1500 and 3000 can bind, and that can be 4500 band. So you can say that this gene like 4000 BP base pair about the gene that might can be digested, and if it is a digested thing, or it might be happen there some RFLP happen like restriction fragment length polymorphism. That's why their band is getting in another position. So in that specific region you can say that the polymorphic band. So hope that you can understand, and it might be monomorphic, it might be polymorphic. In that case I gave that example of 1500 and 3000 and along with the 4500. So these three band can missing and present, and in that case, like that case, you talk about the polymorphism. Like if it is in the one form, it will be the monoformic, but here the 2-3 forms are there, so that's why you can easily understand that it is the polymorphism. So hope that I discuss about the polymorphism, and kind of if you talk about the RFLP, if you want to do the RFLP like restriction fragment, this is the same thing. So before going to that, I want to discuss another thing, how you can run that specific DNA. So in that specific DNA you should go for the Sardan experiment, Sardan blot. So in that you can run the gel. Okay. So sometimes before back kind of radioactive compound may use in that, so that's why it can be taken in that X-ray film. But recently in all the labs are getting kind of do not use that kind of thing, because the radioactive chemical may hazardous, and that's a hazardous thing, so you should not allow to that lab. So that is a thing getting changed in that Sadhan. So that is the thing, and this is a kind of old technique. So recently people don't use that kind of things, because sequencing is there, and also you can digest and also check about that. So why you go for that specific things of old technique? So these are the old technique for exam, but for in real life application it's kind of non-usable. uh, but still any lab can do that if they have their facility of radioactive material handling capacity of that. So they can be, but still it is not possible. So hope that in summary I have discussed the RFLP and ALP along with the SNP. SNPs are important, those are working with the genetics. And yeah, another thing like designing some of the vector, that is also important aspects of, but that is, I think that is not too much serious for the CSIR exam, because if you want to design some of the vector, so you know the component of this vector and what genes are essential in a vector. One day I will in the whiteboard I can explain this kind of what will be the position of the specific gene, and what should be there, and what kind of sequence you might be expected. Okay, this will be there, that means that can give the separate protein, and that can give the some separate structure and stability of that. And one day I'll explain on how to design and how to changes the genome. That's important aspect, and along with that, I want to discuss about the CRISPR-Cas system. That can be a good key tool for the editing of this DNA. That is the another key aspect. Hope that you guys can understand about that. and it's a 15-minute stream already, and in that stream I can also give one update about the CSIR, also decide the exam center where you in the city, exam center city, so you guys can check on the website and can give the feedback. Also you can book some train tickets if it is the center is far away from your place. So hope that you guys understand this stream. In another stream I can I'll discuss about some of these what you can study in the from plant biotechnology or plant biology, because basically those are from this biotech background or microbiology background, they don't have that much of experience in the plant biology. And so in another stream or live stream I'll discuss about that, what will be the plant and how you can remind all those things in the plant. And it's very difficult, I know, because plant things are more difficult, but I'll go give some suggestion on how you can remind those tedious things about the in plant biology. So in that stream, bye bye, tata.